Bananas are of extraordinary significance to human societies. They have been a staple of the human diet since the dawn of recorded history. They are the fourth most important food in the world today, after rice, wheat, and maize. Banana species grow in a wide range of environments and have many human uses, including food, fiber, and as ornamental plants. The banana is of central importance to Hawaii and Pacific societies as a source of food and employment. The Hawaii banana crop was valued at over $10 million in 2007. Banana is a large, perennial, monocotyledonous herb from 6.5 to 30 feet in height. Plant suckers arise from subterranean rhizomes, usually called corms. The flower cluster is an elongated, plump, purple to green bud, sometimes called the bell. As the bell elongates, it exposes semicircular layers of female flowers, then neutral flowers, and finally small, generally non-functional, male flowers. Each group of flowers is arranged in clusters around the stem. Each flower cluster is covered by bracts. About 12 to 20 flowers are produced per cluster. Collectively, the flowering parts and fruit are referred to as the bunch. The individual clusters of fruits are hands, and individual fruits are fingers. The entire above-ground portion of the plant is a false stem that consists of leaves and their fused petiole bases. This is the pseudostem. The pseudostem supports a canopy of 6 to 20 or more leaves. A banana plant can have 200 to 500 fibrous roots. In well-drained, deep, and fertile soils, roots may extend 5 feet deep and 16 feet laterally. Banana fruits are variable in size, shape, and color. They are generally elongate and cylindrical, straight to strongly curved, and about 1 to 16 inches long and 1 to 3 inches in diameter. The fruit apex is important in variety identification. It may be tapered, rounded, or blunt. The skin is thin and tender to thick and leathery, and silver, yellow, green, or red in color. Inside the ripe fruit, the flesh ranges from starchy to sweet, and in color from white, cream, yellow, or yellow-orange to orange. Cultivated varieties are typically seedless. The best elevations for banana farming are from 100 to 800 feet in Hawaii, although the plant will grow well and bear fruit at higher elevations in some locations. From 79 to 82 degrees Fahrenheit is the best for leaf growth, and from 84 to 86 degrees Fahrenheit is best for fruiting and bunch development. The minimum rainfall requirements for banana depend on soil type, planting location, sun exposure, and banana variety. Approximately 80 to 120 inches of rainfall per year is a minimum requirement for an unirrigated commercial banana plantation. 40,000 gallons per acre per week are required to irrigate banana plantations in dry areas. Banana plants can store a significant amount of water within their pseudostems and rhizomes. This enables them to survive periods of drought, although their growth will slow or cease under such conditions. In Hawaii's rocky volcanic soils, bananas suffer significant drought stress and yield reductions after only a few weeks without rain. Banana tolerates a wide range of soils, but well-drained loams are best. Heavy clay soils are not as good for banana. Clay soils tend to be low in organic matter and are poorly aerated. Banana species thrive and grow best in full sun, especially if winds are not high. Banana plants cannot be successfully cultivated in waterlogged soil. Excessive irrigation or poorly drained sites may cause banana mats to float upon their loosely rooted corms. Winds of 25 to 45 miles per hour can topple banana plants. Tall cultivars, such as Valerie, are not recommended for areas exposed to high winds. Steady winds cause significant leaf shredding, drying, and distortion of the crown. Winds cause more damage if the corm is weakened by insect pests such as banana weevils and nematodes. In windy areas, dwarf varieties such as dwarf Chinese and dwarf Brazilian are more resistant to toppling than the taller cultivars. Short cultivars have thick, short pseudostems and compact structure with short, broad leaves. They are easier to harvest and have less green waste for disposal. An ideal growing area for banana would have a banana-free buffer zone surrounding it to minimize exposure to banana bunchy top virus. Please view the additional video on this DVD about banana bunchy top virus. Banana plant growth is rapid until flowering. After the flower bud shoots, the vertical growth of the pseudostem ceases. No new leaves are produced after flowering. Flowering and fruiting occur year-round and fluctuate seasonally. Maximum fruit production occurs during summer and fall. The lifespan of individual plants is about 1 to 1.5 years. Where plants receive care, the lifespan of a banana mat is virtually unlimited because it continually regenerates new plants. Plants produce leaves, flowers, and foliage throughout the year. Individual plants only flower and fruit once. Tissue culture production of the banana plant has become standard for commercial plantations in recent years, mainly because of the advantage of starting a new field with disease-free banana plants. Tissue-cultured banana plants are available from the University of Hawaii at Manoa for a number of banana varieties. Contact the Cooperative Extension Service for more information. Tissue-cultured plants are very small and must be grown in a nursery and sun-hardened before planting them in fields. Bananas are commonly grown from young suckers that are collected from existing fields. Sword suckers have narrow, sword-shaped initial leaves and are attached to a healthy mother plant. Water suckers are those young plants that no longer have a physical connection with the living mother plant. Water suckers do not have the narrow, sword-shaped initial leaves. Sword suckers are preferred to water suckers for planting new fields because of their superior vigor and eventual yield. To create plants for new plantings, sword suckers are not removed during the normal process of thinning out banana clumps, referred to as pruning. Rather, they are saved and reserved for their collection and use as a planting material. Suckers to be used for planting can be given extra light by trimming overhanging leaves and fertilizer before removing them from the mother plant. This increases their viability and vigor. Suckers may be removed from the mother plant when a sucker pseudostem is a minimum of 6 inches in diameter. The sucker is cut away from the mother plant by using a sharp tool. Do not use undersized suckers for starting new fields or plantings. Inspect the detached suckers for disease and pest damage. Care should be taken not to transfer diseased suckers that contain nematodes or other banana pathogens or pests into a new field. If corms are infested with weevils or nematodes, remove the affected tissue, then wash and dip the rhizomes in hot water, 129 degrees Fahrenheit, for 20 minutes, and/or use a 10% bleach solution as the dip treatment. Roots may be entirely trimmed from rhizomes without damaging the plant significantly. Trimmed suckers sprout new roots readily if irrigation or rainfall follows planting. Suckers should be planted almost immediately. The goal of a banana field layout is to maximize efficiency and yield. A target of 600 to 800 production units per acre should be spaced in a manner that allows all horticultural practices to be performed. In Hawaii, tractor-drawn equipment has become the standard for farms that are 10 acres or more in size. Planting layouts range from 1 to 3 lines of bananas separated by a narrow tractor road. In this example, we are looking at a 2-line system on a 20-foot center. The lines are 7 feet apart with a 13-foot tractor road. Williams banana plants are spaced 6 foot in line. The result is 725 plants or production units per acre. In this example, we are looking at a 3-line system on a 25-foot center. The lines are 6 feet apart with a 13-foot tractor road. Williams banana plants are spaced 7 foot in line. The result is 745 plants or production units per acre. The open field is measured and flagged. Row lines are cut and planting holes created with hand tools. Healthy banana suckers or field-ready tissue culture plants are planted. Plant suckers directly into planting holes in a field. Suckers begin to form new roots within days after planting. Leaves emerge from suckers within 7 to 10 days. The survival rate of suckers is about 90 to 100% if they are sufficiently irrigated and fertilized and are free of serious diseases. Bananas are susceptible to a wide range of pests and diseases. A few of them are serious threats to bananas in Hawaii. Some pests and diseases are very contagious and are easily spread. Once established, they are persistent and practically impossible to eradicate. In general, the severity and occurrence of pest outbreaks and plant damage depends on several factors: the environmental conditions, the banana variety, and the specific disease or pest. Wet, rainy environments favor fungal and bacterial diseases. Dry weather or climate favors many types of insect outbreaks such as mites, thrips, and aphids. Some pests, such as scab moths, can cause damage during all types of weather in Hawaii. The highly contagious nature of some diseases mean that many or most plants in a region may become infected by a given pathogen. Although most banana varieties are susceptible to certain severe diseases, some varieties are far more sensitive than others. For example, fusarium wilt is a fungal disease responsible for destroying many commercial plantations of the once popular and widely grown variety Gros Michel or Bluefield in Hawaii. With regard to banana bunchy top virus, which has now spread throughout Southeast Asia and much of the Pacific, the most susceptible varieties are in the Cavendish subgroup: Chinese, Williams, Grand Nain. Insect pests of banana can cause significant damage to fruits and can greatly reduce their marketability. The important banana pests include aphids, ants, thrips, moths and caterpillars, scales, corm weevils, and mites. Banana aphids are found wherever bananas are grown. The banana aphid is a significant pest of banana due to its ability to transmit banana bunchy top virus. Banana aphid populations are often tended by ant species. Aphid excretion of honeydew provides a nutritional and physical substrate for sooty mold fungi, which may diminish fruit quality and physically block photosynthesis. The banana weevil can be very damaging to commercial and home growers. The larvae bore into corms, suckers, and roots. Extensive root destruction is possible and may cause diminished plant growth and yield. Premature toppling of plants and plant death of young plants can occur. A night feeder, the borer can be monitored and trapped by baiting the field with split sections of banana pseudostem. Peel the rhizomes free of lesions and immerse in hot water at 129 degrees Fahrenheit for 10 minutes. Also, a 10% household bleach solution treatment is useful for disinfecting corms. Cover pruning scars on production plants with soil. Do not cover scars from pruned water suckers as they attract the weevils to non-production corms. The banana skipper rolls up banana leaves starting from the leaf margin. Due to the effective biological control of the banana skipper in some locations, chemical treatments are uncommon and largely unnecessary. The banana skipper can be controlled effectively in some locations with naturally occurring biological control insect species or parasitic wasps. The sugarcane bud moth caterpillar is a localized pest that feeds on decaying flowers and causes fruit scarring. The banana moth lays eggs on senescing flowers and on decaying leaves, pseudostems, or fruit. The larvae feed on detritus and decaying plant material. Scales are sap-feeding insects that attack banana leaves and fruits. The coconut scale causes a localized discoloration and yellowing of plant tissue. It is a significant quarantine pest for banana fruits shipped from the Pacific, Hawaii, and to the U.S. mainland. Thrips can scar, stain, or deform banana fruits by feeding on the fruit skin. They also cause scarring during egg laying. Thrips are small, winged insects that feed on banana flowers and/or the tender green skin of developing fruits. The feeding site and thrip species determine the type and extent of fruit damage. Thrips outbreaks can occur during periods of dry weather. Wild birds, bats, and rodents not only feed on banana fruits, but often construct nests within the bunch. Rats can be a serious problem for islanders dependent on bananas. Therefore, bananas should be harvested just before or at the first sign of rat damage. A specified number of hands are allowed to remain on the bunch. The rest are trimmed, with 5 to 6 inches of stem left behind. Sometimes a single finger is allowed to remain below the lowest full hand. As hands mature, remove flowers from the ends of the fingers. When a bunch emerges from a banana plant, spray it with a mild insecticide once per week until it is ready to be covered with a perforated polyethylene sleeve. Now the bunch is ready to cover. Tie a string tightly around the stem about 12 inches above the top hand. Attach a colored ribbon to the bottom for inventory purposes. Bunches with the same colored ribbons are harvested in the same week because they represent the same stage of bunch maturity. This allows consistent forced ripening of all fruits and uniform fruit size. Diseases are major production constraints wherever bananas are grown. The most significant diseases of banana in Hawaii and the Pacific are black leaf streak, caused by the fungus Mycosphaerella fijiensis, banana bunchy top, a viral disease caused by the banana bunchy top virus, and fusarium wilt, caused by the rhesus of fungus Fusarium oxysporum. Fungal diseases cause the greatest pre- and post-harvest production losses and account for a large share of plantation management expenses. Black leaf streak, also known as black sigatoka, is globally distributed and is the most important disease of banana worldwide. This is a highly contagious leaf disease caused by the fungus Mycosphaerella fijiensis. A combination of cultural and chemical practices is recommended. Field sanitation, such as weed control, good nutrition, plants grow quickly, fungicide sprays to kill the pathogen and protect plants, detrashing, or removal of diseased leaves, pruning to remove unwanted suckers, ensuring good drainage and air circulation in the canopy to allow wet leaves to dry, and resistant cultivars. Please refer to a publication on this DVD about this disease. The weekly removal of dead and dying leaves is important for several reasons. First, airflow is improved, which lowers relative humidity. The fungal inocula that commonly infect fruit both pre- and post-harvest often originate on dead leaf tissue, and removing it from the proximity of young fruit is important. Ground-based foliar sprays become more effective because of improved leaf coverage. Visibility is improved. Black leaf streak control is a critical factor in any high rainfall area. Tractor-drawn air blast sprayers are well suited to Hawaii farms. Applications can be made with as little as 10 gallons per acre. Dithiocarbamate fungicides in an oil mix are effective when applied on 10 to 21 day intervals. Triazole compounds are effective with fewer applications per year. Triazole compounds require a fungicide resistance management program that incorporates fungicide rotations and mixing or using fungicides that have alternative modes of action. Panama disease or Panama wilt is a lethal disease caused by the races of soil-borne fungus Fusarium oxysporum. Infection occurs through roots and progresses to the pseudostem. Symptoms are internal stem necrosis, root and rhizome rot, yellow leaves, plant wilting, and plant death. Plants may die during flowering or during periods of moisture stress. The fungus may survive for decades in soils in Hawaii. Management is mainly preventive. Plant resistant varieties or pathogen-free suckers, or use tissue-cultured plants in non-infested soil. Fruit rots are primarily a crown or stem end rot. Crowns are soft, black, and water-soaked. Dark fungal growth may develop in a mass. Stem ends and skin may turn black. Fungal growth may cover the fruit skin with a white to greenish-black color. The pulp may rot and the fingers may drop or ripen. Use good packing house hygiene. Clean and disinfest the packing house regularly. Do not allow rotting fruit to accumulate. Nematodes are microscopic roundworms that live in soils and attack banana roots or corms. The root knot nematodes and burrowing nematode are severe pests of banana in Hawaii. They can weaken root systems, reduce yields, and topple plants before harvest. Infection by root knot nematodes leads to swelling and galling of banana roots. Galled roots may crack and rot. Infected plants can rise from the soil, float, which makes them unstable in high winds or while bearing heavy bunches. High populations can make a field unsuitable for further banana production after 5 years. The burrowing nematode causes lesions to develop in roots and corms. Plants decline, suffer yield losses, and can topple. This pest has a wide host range, including anthurium. Select a nematode-free planting site or fumigate soils before planting. Plant nematode-free suckers or corms, or treat infested material with heat. Banana bunchy top is a very severe disease of banana. The most conspicuous symptom is the stacking up or bunching up or rosetting of leaves. Symptoms can appear on plants of all ages. Bunchy top disease is spread by planting infected material or by insect transmission of the virus between plants. The banana aphid is the only insect vector of the banana bunchy top virus and can transmit the virus by feeding on banana leaves, petioles, or pseudostems. Prevention of the disease is a key component to its management. Ensure the presence of banana-free buffer zones around the farm. Scout for aphids and manage or spot treat them with insecticides. Survey fields regularly for symptomatic plants. Eradicate diseased plants and mats. Use virus-free planting material. Once a region has been contaminated with BBTV, eradication is very difficult. There are no known resistant commercial banana cultivars, and there appears to be more than one strain of the virus. However, in Hawaii, the dwarf Brazilian cultivar is more tolerant of bunchy top than the Cavendish cultivars. For more information, please refer to another video and publications on this DVD about banana bunchy top. The fusion of banana fingers is the result of a genetic mutation or defect, seen particularly in Cavendish varieties. Hands with fused fingers may not be marketable but are completely safe to eat. Remove fused fingers at the time of deflowering and bagging. Individual fingers may ripen prematurely, which could be caused by harvesting overripe bananas, excessive post-harvest storage or transit temperatures, and anthracnose wound infections on fruit skin. Harvest fruit that is at a proper stage of maturity, adequate temperature control after harvest, fungicide dips of fruit, well-ventilated fruit storage. This problem may occur as the result of severe defoliation caused by leaf disease such as black leaf streak or black sigatoka. Senescent spots, a natural part of the ripening process for Cavendish bananas, are numerous, superficial brown flecks on peels. Spots are small and do not enlarge or change color. The brown spots are caused by the death of small groups of cells in the outer peel. Banana fields and plants yield at high level when the number of plants per acre is optimum. For Cavendish varieties on the eastern side of Hawaii Island, a range of 600 to 800 plants or production units per acre can produce over 40,000 pounds per acre of high-quality fruit each year. A production unit consists of a mother plant and a daughter plant. For directional pruning, fields are pruned in a specific direction that is determined by wind direction, slope, and seasonal exposure to the sun. In this field, the healthiest daughter plants that grow on the north or windward side are selected. This provides an anchor for the mother plant, as the bunch will often lean to the south. All other suckers are eliminated. Pruning is done with a sharpened tool that can slice through the corm below the growing point, but above the ground to minimize root damage to the mother plant. Pruning scars from suckers connected to the mother plants are covered with soil to discourage banana weevils. Scars from unattached suckers are not covered with soil in order to attract banana weevils away from the production plants. Nutrient deficiency symptoms are common for banana grown in Hawaii soils. Nitrogen, potassium, calcium, zinc, and boron deficiencies have been documented in several Hawaii banana farms. In some cases, deficiency symptoms may appear as a group because several elements may be lacking. The symptoms are usually both preventable and correctable with choice of location, improved soil characteristics and fertility, and the use of amendments and fertilizers. Crop logging is the periodic analysis of banana leaf tissues for nutrient levels. Please refer to the crop logging document on this DVD for more information on how to select leaf samples for analysis and how to interpret the results. It is wise to also combine crop logging data with soil sampling results to get the best efficiency of fertilizer use. Since fertilizing practices result from crop needs and environmental conditions, banana fertilizers should be designed for each area. After extensive tissue and soil sampling, a fertilizer formula can be custom designed for a specific area. In the Puna district of Hawaii Island, a 13-3-37 blend is used. The slow-release nitrogen has a sulfur or polymer coating to withstand the frequent heavy rainfall. Mature field applications are made with a tractor-drawn bander. Phosphorus levels are low because the media contains a high percentage of lava rock and a low buffering capacity. Potassium levels are high because of crop need and the low levels of naturally available potassium in the soil. Traces of zinc and boron are also included. The average application rate is approximately 1 ton per acre per year. Applications to young fields are made by hand in a circular pattern around the plant. Although banana plants have root systems that occupy a wide cross-section of the soil profile, many of them are at a shallow level and must compete with invasive weeds and plants. Control of weeds is critical for plant nutrition management. Weeds also provide shelter for undesirable insect pests, as well as keeping humidity levels high, which increases fungus activity. Glyphosate herbicides are commonly used to control weeds. Harvesters are usually looking for two ribbon colors during a harvest. One will be the remaining inventory from the previous week, and the other will be the beginning of the next new color. Often the bunch with the older color will be left to grow another week in a known slow section of the field. The younger color will often be taken from faster areas that receive more sunlight. The plant is cut with diagonal slices that allow the plant to be folded over. The bunch is removed in a way that minimizes bending of the stem, which causes point scarring on fruits. Padded trailers with underinflated ATV tires are used to transport bunches to a packing station. Bunches can be hung overnight to reduce the field heat. The banana bunches bagged during a one-week period become the new inventory for that week. With each week having a unique color ribbon, harvested amounts can be averaged over a period of time to provide the market with a consistent supply. Fruit matures on plants quickly during the summer and more slowly during the winter. With Cavendish bananas, the fruit development period, or time from bagging to harvest, can run from 10 weeks for fruit harvested in September to 15 weeks for fruit harvested in March. During the late spring and summer, growers will harvest 5 weeks of fruit over a 4-week period. The opposite is done in the fall-winter months by harvesting 4 weeks of fruit over a 5-week period. This allows farmers to continuously harvest the adjusted amounts 52 weeks per year at a consistent maturity or caliper grade. Yields depend largely upon the cultivation system, species, or variety. For the Cavendish subgroup, given a planting density of approximately 700 plants per acre, banana yields can reach over 40,000 pounds per acre per year. Bunches are de-handed, and the hands are cut into clusters of 4 to 8 fingers. Fruit that does not meet the size or cosmetic standards of a Hawaii Grade A banana is removed at this time. Square, clean cuts are necessary to prevent post-harvest fungus disease. The fruit is weighed on a tray and placed on a conveyor to drain as it moves toward the box area. Each weighed tray fills a 40-pound box and is palletized for shipment to a wholesaler. For more information about banana storage after harvest and bunch ripening procedures, please refer to the document on this DVD about ripening and bunch management practices for banana.